Hello and welcome everybody. This is part of our Regenerative Transition webinar series. This is webinar number 2, Compost Collaboration, Soil Food Web Professionals. So we'll allow for everybody to take a minute or two to join the webinar. And while we're doing that, if you could let us know where you're coming from. So at the bottom of your screen, there is a chat button. If you could click on the chat button and just tell us, oh, hey, Jay's the first one in there from Colorado. Way to go, Jay. All right, we got a lot of USA representing so far.
Here they come.
Yep. Well, we saw some Costa Rica, Netherlands, New Zealand. There we go. There's our international audience. Just had to get past the delay. Coming from all the way around the world.
Yep, I think we've got, yep, all the way around. We've got people we know.
I see Africa representing, Australia.
Cape Town.
Oh, yeah.
More Canada, South Australia.
Fantastic.
More Toronto, Alberta.
Exactly.
Quick tour of the world. France.
You got it. Yay.
Vancouver Island in Canada.
Up in Joe's territory.
Yep.
Okay, well, let's go ahead and get started. Seems like we got quite a few people that have joined so far. All right, so today's topics, we're going to be, again, talking about compost collaboration, and we're going to be talking to some soil food web professionals. We're going to have introductions. We'll take about 5 minutes, and then we'll be doing some talking about on-farm composting by Cory Miller. And then Keisha will talk about the power of microscopy. And then Tobias will be talking about a case study she did with Little Dog Farm. And then we'll have a brief discussion about our Kickstart soil package, and then we'll get into the Q&A. So we'll get some questions from the audience out there, and our panelists will be able to answer that. Our total time for today is about 1.5 hours. All right, so a few housekeeping items here. We will have everybody who's not a panelist in muted mode for the duration of the webinar. Just make sure that we get good audio quality. We usually have hundreds, if not thousands, of people attend these. And then I want to describe to you about the Q&A section and the chat buttons. So if you want to post a question for the panelist at the end of the webinar to answer the questions that you have, make sure that, again, at the bottom of your screen, there's a Q&A button. Type your questions for panelists in the Q&A section. Then we also have the chat button, which a lot of you have already used, and this is so that you can have a good dialogue in the background with other attendees. So chat amongst yourselves while we're actually doing the webinar. That's great. But again, if you put a question that you want for panelists in the chat window, it will probably get missed. So make sure that if you have a question for the panelists, go ahead and put it in the Q&A section. And then other than that, please just go ahead and enjoy. So let's talk a little bit about our Regenerative Transition webinar series that we have here. We've already had the first webinar, which was Regenerative Vision, Soil Life and Global Communities, and you can watch the replay at webinar.soilfoodweb.com or at YouTube at Soil Food Web School. And then today is our compost collaboration webinar. And then we have a third webinar, Dirt to Soil, Transitioning to Biological Farming. That's going to be 11 a.m. Pacific on Wednesday, November 1st. And we'll talk with a number of practitioners and what we do to help our clients transition from dirt to soil. All right. Now let's go ahead and get introduced to our panelists. So Elaine, why don't you go ahead and introduce yourself?
Okay, so I'm Dr. Elaine Ingham. I've been working on the soil food web for the last 45 years of my career in academia. In the early 2000s, I'm kind of converted over to do more of a commercial situation where we taught just the general public. You didn't have to have a master's or a PhD to come to work with us then. And we've been getting this information out to people so that they can increase their yields. They can have better flavor, better nutrient conditions in the food that you're eating, your animals eating the same good food all the way around. It's a really huge difference that now compared to 45 years ago when I walked into all of the soils people offices at Colorado State University and outlined a work that I wanted to do for my PhD, which was to look at all these different kinds of fungi and figure out what they were doing in soil, because the attitude at the time was they didn't do anything. They were just sort of there. And we all know Mother Nature doesn't work that way. If they're there, they're doing a job. And bacteria and fungi, protozoa, nematodes, microarthropods, yada, 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 they do an immense amount of work for your plant. So we've got to get these organisms back into the soil. And that's the only way you will get away from pesticides, inorganic fertilizers, having to irrigate all the time. Oh, the list is getting longer every day that we continue to work on what do these organisms do for your plant, and then vice versa. What does the plant do for your organisms? It's a bootstrapping system to leading to better and better production. So hope I didn't take too long.
Not at all. Thanks, Elaine. That's a great introduction. Cory.
Cory Miller, Missoula, Montana. Have Grass Valley Farms, and growing little operation. Started 1,000 acres. We're up over 3,000 now, and farm primarily sod, cattle, and hay. We don't ship anything outside of our local community, so it's kind of a claim to fame to keep it local. But growing quick and expanding every day, and a lot of that is through Soil Food Web and learning everything that I can about biological farming. And I'm just glad to be here and to be able to share that story that we're doing.
Great. Thanks, Cory. Can't wait to see your presentation. All right, Joe.
Hi, everyone. Thanks for having me here today. My name is Joe Tobias. I'm based in Vancouver, BC. I operate a company called Root Shoot Soils, which serves on the unceded territory of the Coast Salish peoples, including the Musqueam, Squamish, and Tsleil-Waututh nations. I work primarily with land stewards, farmers throughout British Columbia, Alberta, Ontario, and Saskatchewan. Thanks.
Great. Thanks, Joe. Keisha and Casey.
Oh, I'm unmuted. Okay. I'm Keisha Ernst. I own a compost company called Catalyst Bioamendments with this guy, and also a consultant. I basically think of myself as a microbe farmer. I get into almost anything you could imagine that you'd be doing with a microscope and soil.
Casey, and we also have a consulting company where we do on-site consultant with farms, kind of converting them away from chemical use or just integrating biology into their systems, and that company is Soil Matters.
Great. Thanks, Keisha and Casey. And I'm Brian Vagg. I will be your host today. I'm a Soil Food Web consultant. I have a consulting company called Sprouting Soil. And then, as you can see, Keisha, Casey, myself, and my wife have also combined forces, and we've created a consulting company called Soil Matters. And this is really allows us to be able to tackle even larger scale kind of operations. So I think the future is bright for all of us. Okay, let's go ahead and move on. So, Cory, take it away.
Thank you very much, Brian.
Mm-hmm.
So some of you, if you haven't seen it, I was on here 6 months, 7, 8 months ago now, maybe, and did presentation part 1, and it was a lot of the details on how we got started. But I wanted to, this is kind of second part, so what we've learned and where we're heading, and then actually being the end of the growing season, kind of what our results were. So we own 700 acres. We lease another 2,400, and we're growing fast. And I think, the main reason for that growth is the biological farming that we're doing. It's very easy to tell this story to landowners that aren't effectively utilizing their land, and us being able to get in there and use these practices, they like it. And so as a producer, and being able to talk this language and understand, and to communicate to them that we're really benefiting their land, the changes that we're making is really helped us grow. And so I want to make that point that, it's easy to tell that story. Currently, as of today, we're managing about 3,100 acres, and all of it is within 3 miles of the city of Missoula. And that's kind of a big deal because we're really close to all of our customers. Our beef, when we process it, it doesn't travel more than 10 to 15 miles through this process of getting it from being born to somebody's table. And our customers here in Missoula, are really progressive town, and they like that. So when I talked last spring, it was a lot about statistics and what we had. And so I just wanted to share kind of some updates. So 21 to 22, we had a 22% increase in production. 22 to 23, we're almost 40% over that increase in production. And a lot of that, we had very favorable weather conditions this year, a wet spring, hot, warm weather throughout the summer, and that was beneficial for production. The other thing we did is we wanted to take some of the hay that we produce off before the grasses went to seed so that we could continue growth of those grasses. And so we made some haylage bales at the end of May, early June, which in our climate, that's tough to do, or at least to get hay off that early. And we were able to do that, and it was very successful. You know, a lot of people don't know what haylage is, and they don't know that it's very good for your animals because it's partially broken down. It's, you know, when we talk to horse people, we talk to it about being a product that is half the digestion has already happened. And so the effort that it takes your animals to convert that feed into energy is easier for them. And so, yeah, product out, we sent it out to sample to a bunch of our customers, and of course it's all gone now. So we're excited. It's opened up a new market for us, and will be some growth that we can do in a time when we couldn't make hay before. So yeah, on the hay side, very good production this year. Meat side, 21 to 22, we grew fast. 23, our problem became production, or at least processing of animals. As everybody knows, across the country, really hard to get the dates for animal processing, and it's continued on. A lot of farmers are choosing to go the direct marketing route, and so it's made bottlenecks in our small processing facilities. Things are looking good in 24. We've got dates now for the entire year, and we're big enough now where we can fill those dates. So a lot of growth. We doubled the size of our herd, and are always sold out of beef. So really good for us on the beef side as well. Other thing that we do here is we grow sod. And for me, I looked at it as an opportunity for us to have a cash crop that was very valuable, because we're surrounded by subdivisions. And I know none of you guys, or at least other panelists, have been to our place. But it is, I mean, we look out of my office at $1 million homes and golf courses, and it's just development everywhere. And it's what can we do to keep it, or at least take advantage of that. So sod was one of those. Of the 700 acres of the main ranch, we put 56 in sod. And you know, it's really interesting. If anybody's been to Missoula, there's a place called Karis Park where all of the events happen, concerts. There's a pavilion, a stage. There's a whitewater rafting wave called Brennan's Wave. Very popular, probably 5,000 to 10,000 people a week visit this area. And we got the contract to resod that this summer. So that was a big deal. And we got to put our signs up, and our sod sales took off at that point. And so a good way to get a new business going when you're changing directions. Excuse me. I wanted the main part of my presentation today is kind of talk about what I've learned along the way. And I was a soil food web school student, I think going on 2 years ago now. But we make a lot of compost here on the ranch, and we also source some. And so that's how I was able to meet Casey and Keisha. And we got some of their compost originally, I think 2 years in a row now. And it was a very good product. And we use that as an inoculant to get a lot of our stuff going when we were first starting out. So we make, you know, our base compost in a thermophilic way, which is what I call the Dr. Elaine style, or what I learned in her courses. But when we store it, we found some very useful stuff in the Johnson Sioux and what the Dr. David Johnson talks about of developing heavier fungal. And we do some things on our ranch that are very important for the breakdown of lignity material. So we bale graze our cattle in the wintertime for putting more nutrients down on the soil. And what we find in the springtime, that it's very difficult to get those fields cleaned up. And so we store compost for, you know, we have some now that's 3 years old. but very, very high fungal. We're feeding it every day. We're watering it every day. We baby this stuff. But what we found is it gives us that super high fungal that we're able to go and spray and or make into a liquid, and in the springtime go and put it on this leftover litter where we bale graze cows. And in 3 to 4 weeks, those areas are cleaned up and new plant growth coming through. And so, you know, everybody asks why we go through the effort to do it, because it keeps us from having to till the field. It keeps us from having to mix this material into our soil. And was something that we discovered last year as we were cleaning up. You know, we use the new stuff, or are we going to use the old stuff? And we tried both, and the old stuff we found more fungal, and was surprised me in its ability to clean up that lignin. Sorry, move on again. When I talk to people that come out to the ranch for tours, we talk about making our liquid extracts, and I call them a soup. I refer to it as our soup, and our compost is always the base for that soup. So the biology doesn't matter what we're doing to any crop that we're growing on the ranch. Everything starts with our biological soup. That's an extract from compost. We keep at least as far as our really good stuff, the previous 3 years worth. So stuff that's 3 years old, of course, higher fungal, we're feeding it. But our new stuff, you know, more bacteria, more protozoa, really good nematode action. That's what we're using to seed coat. That's what we're using for our soil drenches. And we mix these just according to what shows up on the microscope. And I'm so excited that you guys get to hear Keisha today talk about that, because you know, the importance, what I learned in the soil food web school was the ability to use a microscope and identify what's in the soil. And it makes it so easy. I mean, I'm sitting here right next to you guys. I have my lab, and I can go and pull a sample at any time, any day, and see what we're doing, where we're going, and make plans. And yeah, I'm excited you guys get to learn that. But with this soup, we have to add stuff to it. So this is our extractor setup. This is what we use to produce our liquid, but it also gives us the ability to inject others. And a little story that I want to tell this year, you know, we knew we were missing something. I'm very goal oriented and production based, and I wanted I knew that we could really push the limits of what our land was capable of. And so for 2 years, the first 2 seasons, it was biology, adding biology to the soil, doing everything we could to get our soil working at maximum production. But I knew that we were always missing something with our plants. You know, there was always, they just weren't perfect looking. And you know, not only do I study Dr. Elaine, but I read everywhere I can, just like I'm sure all of you do. And of course, found out about plant sap analysis testing and how it worked and what it was. And so this year we decided to try out some plant sap analysis testing. And it's really, I mean, think about, you go to the doctor and they can't figure out what's wrong with you. They do a blood test, and that blood test tells you basically statistical level, or not statistical, but actual levels of mineral, whatever it is in your body they're trying to identify. And with that, you can then make an informed decision going forward. So we chose to do that with our plants. And it was really interesting to see, because most of the data for this is compared to conventional farming. And so there's not a lot of data out there for people that are doing sap analysis as well as straight biological or straight organic farming. And this was performed this season on all of our irrigated fields, and really was, when we started in May, was to get a baseline. So we let everything grow up, and just before our first cuttings, we went out there and took samples. And I will tell you, it's a very tedious process. You know, you're pulling a plant. So alfalfa plant, for example, we're pulling trifoliate leaves off the top and bottom of the plant, so new and old leaves. And then we're keeping them separated and then sending them in to be analyzed. And what's interesting is the data that it gives you. And so by looking at our plants, I was assuming that our health of our plants was weak, that we were overusing our resource. And what I found was really the opposite, that we were excessive in a lot of minerals. We had some micros that we were deficient in, but it answered the question to me of, is this working? What is the biology doing? I had other evidence to show that we were improving the soil, but the SAP analysis really told me that what I was doing in the soil was now making the transfer to the plant. And that data allowed us to make some really informed decisions and also explore some new research. So as you can see, and I'll just kind of go over this real quickly, it was here for presentation only, but anything that's in the green, blue, or purple is higher than average, higher than necessary. If it's red or yellow, it's below. So you can see that we're really only low in some cobalt, some copper, some zinc, and some iron, which that is really easy for me to chelate and get put onto our crops, mixing it with what I call our soup. And so I want to just go one more. We use the results to target specific deficiencies that we found in this testing. And it opened up some discovery into more Facebook and learning what other people are doing. And so helped get a group kind of put together on Facebook called Farm Made Bioferts. And for information purposes, it is a great place to go and learn about what you can make. We make super magro, which super magro is a way for us to chelate mineral, and then we're just adding some different forms of mineral and using biology to chelate that, and then putting it with our compost extracts and out onto our plants. And we had significant, or I shouldn't say, I'm not going to say significant because I really don't like that word, but we had noticeable improvements in just how our plants look throughout the growing season. Our testing, every time we did it, came back better and better. And very simple. I mean, we're putting maybe 3 to 5 pounds of chelated iron per 10 acres on our place. So very little as far as what it was needed, but it was applied in a way that the plants could use it immediately and make those improvements. One thing that we learned through this process is the use of clean water. We have an RO system on the ranch for making these extracts, and incredibly important for getting that mineral into the plant. We had performed the plant sap test just a couple of days before cutting or grazing. We'd get the results back, and then we would target our treatments 10 days or so after the cutting or grazing, so right when leaves are starting to show back up. Another thing I did is I would be out all night spraying, and it was hard to do, so we went to Harbor Freight and put huge light bars, so I looked like the sun cruising around in the field at night, but found that it was a very effective way to get the plants to absorb these minerals that we were putting out. Moving on, looking back at what we did, we know that by targeting applications of the nutrients I need to increase our production, it was obvious here. We were up 40%, even better than with biology. But what was really interesting to me is talking to AEA and John Kemp's organization. They were really surprised to see how good a shape our soil was in with the conditions. Their consultant Sean was out here, and they really, they're very smart people, and they've done a lot of this on a lot of different kinds of crops and fields. But they were surprised to see how effective the biology was in our soil. They just didn't expect and didn't know how to make a determination based upon what our SAP test showed. And for me, that was I kind of walked away patting myself on the back that, okay, what we're doing is really good. And they don't know it, but our biology is what's making them look good. And I think that happens in farming a lot. If you're in a climate that has, good water, timely water, you have biology doing the job, you don't realize it. And I think a lot of us as producers overlook that. And we overlook the capability, as Elaine was saying earlier, of what biology does. And we're finding out more and more every day about what its capabilities, what it can do, how it changes, how it improves water holding capacity. I can tell you from being a producer on this side, I've seen all of that. We're just finished our third growing season, and I've seen this change incrementally happen, almost weekly. I find something new. Our sod field, we're not using any synthetic or fertilizers on that sod field at all. We're making everything ourselves. And when you go out there and you see mushrooms growing actively in a sod field, people wonder why. But for me, I know that we're building soil. And when we roll up sod, we're taking just the thatch. But what really makes our sod special is that on the outside of that roll of sod, you may count 400 or 500 worms that are in the netting on there. And we did an install last fall, almost a year ago now. It rained an inch the next day, and I went out to see the customer and, how's the grass look? Is it floating away because it's so much rain for us? And she said, I don't know, I can't go out of my house. I'm scared of worms. And her entire driveway was covered with worms that came out of our sod. And I said, you go make sure that those all get back safely. They're going to continue to improve your soil. And, made a believer that worms are okay. And now really good looking grass in her front yard. And I think it's just the knowledge. People need to know that the stuff under the soil does a very beneficial job for you. And it's much easier to work with it than try to work against it.
Awesome presentation, Cory. I can't wait to see next year's, the 2024 data.
Yeah, we have some. We're early. The season just finished, so a lot of the testing that we've done. Everybody's heard of the Biggest Little Farm show, and one of the scientists that works for that organization. I didn't realize this, but just down the road from me, they have a research ranch basically, and they have a Swedish scientist there, PhD gal, that is doing mycorrhizal relationships to weeds and other plants. And they found that some really cool research there. And so she asked me to be part of a study. And so they were out monthly taking samples of these inoculated seeds that we had done. And throughout, so I think we did some corn, some alfalfa, but she's also done it with 10 other farmers in Montana, kind of all across the state. And it's going to be really interesting in February, March, when they get done doing all the analysis, to see if our inoculants really help, because each of us kind of did something different. And that's going to be really exciting, because I'm confident that, what I see in the field and what she's going to see in her in her science is going to be really interesting. Not only for me, but a lot of other people that really enjoy this stuff.
Fantastic. Can't wait for it to be shared. Awesome. Thanks, Cory. All right.
Yeah.
Let's move on. Keisha.
Thought I was after Joe. I'm unprepared today, you guys. Okay, so usually we're on here so often, and usually when we do these webinars, it's all about what a day is like in the life of us at our job. And I've presented that so many times that I thought, try something new today. So I've got just a little bit of time, and I realized when I was timing this out, it's a big topic, but I think it's worth speaking about here. And it's actually how we build our compost recipes using the microscope. So, everybody's going to hint, everybody that's here today uses microscopes to look at their soil, to look at their compost. And that's pretty normal and known. But what I want to talk about is actually using the microscope to make a product that's better than BioComplete. So it's like the BioComplete numbers, that's our minimum, right? That's what we're all going for when we make piles. But if we actually assess every single windrow when we're done, and we've written down everything along the process, we're able to alter our recipes to make them even better than before. Like, for example, right now in our lot, we have 3 windrows. They all have the exact same materials, but they're put together differently. They're different ratios of materials. Now, the whole time they were composting, they looked really similar in the way their temperatures were rising and falling, even in their texture. Now that they've come down to their, they're at the ambient temperature of outside, and I look at them under the microscope, I found one of those windrows is far, far, far superior to the other two. And so the next time we make compost, you guys want us to make windrow number 3, not windrow number 1 or 2. So we're going to go back to all of our notes. We're going to see what was it that made that windrow so special. And we're going to try to mimic that the next time around. And as we move through time using our microscope as a tool to actually see these invisible creatures that we're working with, we're going to be able to improve our processes over and over again. Thank you. New buttons. I'm actually pushing myself forward. I don't have to ask Sammy. All right. So what's living in our compost? A lot of folks, they identify as microbe farmers. They're doing everything they can to make a compost pile that is beautiful and full of fungi, bacteria, protozoa, nematodes, microarthropods, and everything else. And, I'll hear, I know it's good. I can see the fungi growing in the pile. I know it's good. Well, often when people can see the microbes on the compost, it might be a type of mold that isn't really the fungi we're looking for. And I had a photo here. This is an example. We were at our compost lot. We saw this gorgeous fungi. It's ropey. It's holding the compost together. We didn't just high five and say, yeah, like we just did a great job. No, we took that chunk and we pulled it directly into the lab to look at it because we want to check ourselves. My eyes say it can see fungi, but can I see nematodes? Can I see amoeba? Can I see diversity? No. So the microscope is the tool to help us figure out what's actually living. It would be like being a cattle farm without being able to see your cows. The microscope is the tool. All right, so what do you do? If you look at your compost, you've been working really, really hard to make the best microbially diverse compost, and you look at it under the microscope, and this is what you see. This has been a reality for a lot of the clients that I have. They've really put their all into their compost pile, and then when we look at it, there's not even really organic material present in the compost pile. And it's like, well, how do we get to a place where we've tried so hard, yet we only have bacteria? So let's try to answer these questions very, very quickly in this presentation. So as we're going through making our compost piles, we have to realize that the microbes are not an inoculum that we buy from somewhere else. If you're going to buy an inoculum for a compost pile, it's best to use compost that's already been verified with a microscope. Or we like to have parties where we teach people how to make compost. Everyone goes home to make compost. Then they bring a little bit of inoculum back and we all share it. That's the best way to get free inoculum for your compost pile. Second best is just collecting lots and lots and lots of diverse organic materials to put into the pile. So your wood chips, all of your different species of plants, even your hands have fungal inoculum on them. The whole world is covered in microbes. So the more diversity that goes into the pile, the more diversity you're going to see on the other side when it finishes. Now, the other thing is you got to write everything down. Because even if you've sourced the best organic materials in the world, even if your wood chips are absolutely just teeming with fungi, if you don't write everything down during your processes and say your pile gets overwatered at some point and is stinky for 4 weeks and you don't write that down, you've made 15 different piles. You go to look at what happened to my microbes. This one pile is bad. You're not going to be able to know because it's difficult to remember all these subtle little nuances. I mean, we'll go as far as to even write down who we got our wood chips from, what species we thought were in the pile of wood chips. We might write down how we store our materials. Like I go as hard as I can on keeping data. We keep notes on whether there's flies present, whether we see anything visible on the compost pile, like that fungi that I showed the picture of earlier, I would put that in my notes. And then after we're finished building the pile, we look at it with a microscope and we see, did we do a good job? Did we do a bad job? Either way, you want to know why you're in that situation. If you need more fungi, if you need more nematodes, if you need protozoa in your pile, you want to be able to go back through and figure out where it was that you went wrong, or again, where you went right. That happens too. Sometimes we'll have the best compost pile, that I've ever seen, and we just didn't take thorough notes. And I'm going, why is this one better than the other 3 when all of the materials were the same? And usually it's ratio or treatment, ratio of recipes or treatment of the pile that'll give you that answer. Okay, so fungi. Fungi is the one, I get this question more than any question out there. How do I get fungal diversity in my pile? Where do I buy my fungal inoculum from? Should I use oyster mushrooms or garden giants? Or, what kind of fungi is going to benefit my pile? And, we've tried buying, sourcing mushroom blocks. In the beginning, we were doing everything we could to grow as many different species of fungi as possible in our piles. But honestly, those local native species, the species of fungi that are thriving in your local area, those are your best bet. And those are going to be covering, dried leaves. They're going to be covering wood chips. Like I said before, fungal spores are they're flying through the air. They're on everything. I mean, a really good aged wood chip pile, you can put a wood chip under the microscope, shake it up in your test tube. You're going to see fungal feeding nematodes. You're going to see protozoa. It's going to look like a diverse environment, literally on an aged wood chip. So if you're starting with the right materials and then you treat your pile well the whole way through, you're going to see these organisms growing on the other side, and they're going to be happy. Really, it's diversity of input materials that's going to be the key the whole way across the board. And so with fungi, wood chips, that's their food source, right? Cory brought this up, 60% of the pile is wood chips. So this is a place where you can get a lot of diversity for your pile without too much work. I mean, as far as storing wood chips go, you don't have to put them inside a building. You don't have to keep them dry. They can actually just sit out in the elements, and it's even better for them. As the wind blows, as critters crawl around on them, they're going to keep collecting diversity of fungi, and it's going to start growing. Protozoa. A lot of people come to me and their protozoa numbers are low. They've got fungi, they've got bacteria, the structure of the pile is there, but they're just not seeing their predators. And so like protozoa are like little wolves in compost. They're hunting all the time, and they need it to be moist enough to reproduce and to live. And they need a good food resource. So oftentimes when we're not seeing, in our windrows, this happens all the time. Compost gets dry, I see protozoa numbers dip. Right away, they start to fall. And so I know when I look at my samples, how moist the compost pile is or how many greens we put in. So if you're not seeing a lot of protozoa in your piles, you're gonna wanna look to your input materials, and you're gonna wanna gather as many fresh greens as possible. You think that our microorganisms are only in the soil, but that's not true. They actually cover the whole plant. And so, I hear you even find nematodes up on the tops of plants. They can cruise around. So remember that your input materials in all three categories need to be as diverse as possible. We love making compost with friends, because the more hands we have around, the more eyes we have thinking about diversity, the more that we're going to get out of the situation. A lot of people don't see weeds like plantain or mustard or dock as an input material. They think, I'm just going to add grass. I'm just going to use alfalfa. But for me, I want to see hundreds of species of green material in my compost pile, especially at the beginning. Once you get your culture of microbes growing, you've got all your protozoa and they're nice and diverse. They can eat lots of different foods. But I say in the beginning, you want to invest as much time as possible. And doing it with friends, it just makes it, many hands makes light work, right? All right, then nematodes. So nematodes are the funnest. This is something that we commit to over time. And I have to recommend nematode extractions. If you don't know how to do a nematode extraction, look up Behrman funnels, or just type it into Google, it'll teach you. But then you get to see all of the nematodes in your system. And so what's really important when you're talking about nematodes is also they're like worms, they're roundworms. They need moisture, just like protozoa, just like fungi. But really they need diverse food resources, and they need structure in the compost pile so they can move around. If you see them under the microscope, they really wiggle. They can jam around that slide so fast, it's easy to lose them. And so they really need that structure that your fungi is providing. In the beginning, what you're often going to see is a lot of bacterial feeders. They can reproduce very quickly, and often you won't see a huge diversity. But over time, as you're adding more and more fresh input materials, right, the more different green species we can get, the more wood chips you're going to be seeding in these different nematodes. And over time, you'll start to see your bacterial feeders. Some of them will have big fancy lips or what looks like crazy hoods, or there'll be different shapes, different sizes. Soon you'll start to see fungal feeders show up. I remember when we saw fungal feeders on our compost, it was just like, oh, we freaked out, because we've been waiting for like 6 months for them, or just every single time we look, it's just bacterial feeders. Well, now after 5 years, I'm happy to report when I do a nematode extract, I see predators almost every single time. I even have clients report back, I saw a predatory nematode in your compost. And to me, this is like, it's like having wolves populating my compost. And it took a long time to get there. I remember Elaine telling me some nematodes, they don't just have babies every month. Some nematodes can take up to a year, and maybe they're only going to have one baby at a time. It's like people. And so we need to hold these conditions in our compost piles so that our nematodes can thrive and take the time that it's going to take for them to reproduce. They're not like bacteria. They're not just splitting every chance they get. They actually have to go through the whole process of having their young. And so, yeah, this is something that you just kind of keep, I think of it as like collecting trading cards. And eventually you get to this point where you just have this, rich nematode landscape that's really fun to look at. I will say that this one photo that I have right here where they're all grouped in together, that's a nematode star. I show this picture every chance I get. This is a bacteria that actually, it doesn't care what nematode is present. It wants to eat them all. It happens in flooded conditions. And so it's just kind of hanging out in the soil. When the flood comes, it makes the tips of their tails sticky. And then as the nematodes swimming around, their tails kind of catch together and then they thrash as a big ball. These guys just keep moving and collecting nematodes until the flood goes away and that bacteria eats them. Super crazy. If you do nematode extractions, you'll see this happen. We come across this all the time now. It was like a magical mystery. It took us years to figure out what was actually going on. All right, so let me move this. So just to wrap it all up, the microscope is going to be your most important tool. If you call yourself a microbe farmer like I do, you're going to need that microscope. You want to be able to see the creatures that you're farming. You're going to want to be able to learn as you go and create better and better compost piles and understand when your compost is actually taking in your soil. It's amazing to be able to see protozoa in your compost, water that soup, as Cory would call it, into your crops, and then look at that under a microscope and see your nematodes and your protozoa and your fungi taking over right there in your growing space. This tool it's impossible for me to really let you know how important it is to what we do, and also how fun. I prefer looking at a microscope to sitting and watching TV. So the other thing to realize is even if you have all your microbes in place at the beginning of your compost pile, you've got to keep the conditions throughout. As microbe farmers, that's basically what we're doing. We're doing the best we can to get everybody into the party, and then we're trying to keep those conditions so everybody has a good time. If it's overwatered, if it's underwatered, if it's too compacted, we can smash out all that life easily, and we will not know we've done it unless we use our microscopes. So the last thing is we've got to be a detective. Keeping track of your notes, really paying attention to what you're doing and reflecting on what happens. Whether you're winning and everything is going great and you've had perfect luck as a consultant, or whether you're trying and just not hitting the mark, you want to know why it's happening. So write everything down, do your best to be a detective. And I like to always say, just don't take it too seriously. Just get started today. Start trying and learn more as you go. It's a process. You don't have to know it all right away. And thank you for listening. Thank you for the hearts and the little emoji clappies. So cute. Appreciate you guys.
As always, Keisha, fantastic presentation. I love the pictures in there too. Good job. Okay, Joe, let's go ahead and have you take it away.
All right. Thanks, Brian. So I'll be presenting Little Dog Farm as part of today's webinar. So these are my farmer collaborators on the right. That's Kathy and Ian Finley. They are the owners of Little Dog Farm, which inhabits and learns and grows on Ktunaxa land. Their regenerative farming career started back in 2013 when they moved their family to a 5-acre lot in Langley, BC. After 3 years of farming, they were supplying 11 local restaurants, providing 40 CSA boxes to regular customers, raising heritage pig breeds and grass-finished beef, attending 4 farmers markets, and basically hosting events every weekend. In 2015, their farm was awarded a Chamber of Commerce Award for Sustainable Business of the Year. And in 2020, they decided to move from their 5-acre lot to a 60-acre parcel of land in Creston, BC, and they've been busy reestablishing their farm business there in Creston. Okay, so here's a quick summary of my first conversation in 2020 with Ian about the new farm in Creston that they purchased. Their goal is a mixed species orchard for cider production. Soil health is an issue. Ian dug trial holes and found virtually little to no soil life. Poor organic matter levels on 2 fields that they do want to turn into orchards. Baseline values in the beginning were 2.2% and 3.8% as of March 2021. Pioneer weed species everywhere. So Ian wants to produce compost extracts and teas on site. They also want to do soil testing for the initial 5 years until the orchards are established and flourishing. Community building is also very important for them. They want to build a restaurant linked to the farm, showcasing produce from the farm and other local suppliers. They are in the midst of building a facility right now where they'll be processing their apples for cider production. And they also want to make the farm an educational hub where people can learn about regenerative agriculture, nutrient-dense foods, and soil health. So in April 2021, they invited me to their farm to do the initial site visit. These were the photos I took during that time. Soil management, historical data does not exist. The former owner said he did not spray. He primarily grew hay and tilled heavily for a significant number of years. And the land was left bare when Kathy and Ian took ownership of the property. So you can see just by these photos that a lot of work needed to get it done to a living state. So here is what basically for this one, May 2021, there wasn't much really happening at the farm. It was just, you know, they seeded cover crops, nothing was germinating. So we decided to till. We said, you know, we got to break up that compaction. There's nothing much for us left to do at this time, but let's try to get some equipment out there to make sure that we can break up some of these clods, basically. I don't think I'm working these controls very well. Sorry, everyone. I'm skipping some slides. But anyways, here's a different section of the farm. You can see how there's very little structure in the soil. There's an area also where they planted vegetables on the very right-hand side. These carrots were planted in the fall of the previous year and showed no signs of decomposition, so indicating the absence of biological activity. So Ian set out to build compost. This was his initial setup. Each of these cylinders can hold about a cubic yard of material. We started with roughly 4 cubic yards for 25 acres, which is the size of roughly the estimated size of their fields that they are turning into orchards. Now, the reason for choosing a small volume to begin with is so that Ian can get a sense of how to properly manage his piles and have a good understanding of the resources available to him locally, where and when. And this is our recipe that we worked with. Beer mash or spent grain and chicken manure made up our high nitrogen material, grass clippings, weeds, lots of different types of weeds they had on the farm, hay, silage made up the green component, and wood chips for brown or woody component. So I opted for a higher than normal ratio of foods that favored bacteria because of the state of the soil. We needed to rebuild the foundation from scratch, as you saw with the photos earlier. And it was important to build micro aggregates. So we wanted to take advantage of bacteria's ability to multiply exponentially, produce their secretions that promote the binding and restructuring of soil particles. And throughout the month of May, Ian recorded the temperature from each of his compost piles, took photos of the logs, sent them to me, and I supported him remotely in managing and monitoring his piles. So this data allowed us to identify when is it a good time to turn the piles, or why is this particular pile behaving differently than the others, even though the same ingredients were used. So there were some microclimates that we needed to consider, and moisture levels as well that we needed to factor in as part of compost management. And this data also helped us improve future recipes. So taking notes, as Keisha mentioned, of what ingredients you're using and how that compost progressed over time. Out of the 4 piles, we were only able to use 2 piles for our treatments. The other 2 did not, unfortunately, were not successful. So we were 50% successful with our thermophilic composting in the beginning. So here's the raw data Fungal to bacterial ratio is below the minimum desired for deciduous trees. So this is a highly bacterial dominated compost, but on purpose. So the question is, can we still use these piles for extracts and teas? My answer is absolutely. We could use whatever we have. So this is what Little Dog Farm had to fix on the left. The compost piles had a very good population of bacterial, basically bacterial feeding nematodes and protozoa. So both of these groups have species that feed on bacteria, and having those predators in place keep the bacterial communities active. They're forced to reproduce, they're forced to multiply and build habitats to protect themselves. So the reproducing, multiplying, and creating habitats in the soil maintains the process of decomposition. Which results in increased soil aggregation. So the bacteria promotes decomposition, and the byproducts of decomposition helps the formation of new structures in the soil. So we are rebuilding microaggregates. So studies have found that the stability of aggregates is partly due to the long chain sugars that produced by microorganisms decomposing plant residue trapped in the aggregate. So as Walter Yenny liked to mention, soil microbiologist based in Australia, he likes to say, add nothing in the soil. And what he means by that is to basically promote complex nothingness, have more pore spaces, increase the void in your soil. And the way to do that is to restore microbial activity that produce enzymes that break down organic matter. So one thing to also keep in mind is to combine organic matter building practices, such as cover cropping, with your liquid inoculants to rebuild the soil structure. So taking a look at the data from a different perspective, these are flagellates, organisms that feed on bacteria. The red bar refers to the minimum desired. So both piles that he built back in May of 2021 exceeded the minimum. And here's amoeba, another group of organisms that feed on bacteria and other amoeba as well. Both piles will wear well above the desired numbers. And then we have the nematode numbers. The green is the minimum desired. We did not have predatory nematodes, so nematodes that fed on other nematodes, but we did have other free living nematodes, and they were well above the minimum average. So it was also in 2021 when we had the heat wave here in BC. We were not able to treat the fields until October of that year. We had to wait a few months before we can actually do the work. So in October, I revisited the farm and trained Ian on how to make his own bioinoculants, his extracts and teas, using the compost piles that he assembled 4 to 5 months before. So using microscopy, supporting him in quality assurance, making sure that the organisms in his compost were making their way out of the sprayer. At this point, the volume that he was spraying was roughly 25 gallons per acre, which can be considered high for most people. But again, with the state of the soil, we needed to really pump up that biological activity. So this is the North Orchard section of the farm. On the left is what the field looked like in May 2021 during my initial visit. And exactly a year later, after only 3 treatments, the cover crops that didn't germinate woke up. And this is what it looked like on the right-hand side. This is the same field a few months later. You can see how much biomass has been created after just 3 treatments and sufficient moisture. This was our first step in rejuvenating life in the soil. And Ian's a pretty tall guy, so that rise quite impressive. At this point, it was just allowing the plants to do their thing, allowing them to set seed and reseed for the next season before preparing the field for tree planting. So really encouraging above ground growth that will eventually get incorporated into the soil to nurture below ground growth. Here's a different section of the farm. So this one is called the South Orchard. On the left-hand side is what the soil looked like during my initial visit in April 2021. And this is the section that they started working on first to prep for tree planting. Because the majority of the cover crops did not germinate initially before the treatments, we discussed the role of weeds. Weeds essentially became the covers that we needed to rebuild soil life. So whoever wanted to be there, we let them be there. So was it necessary to buy more seeds and take that financial risk if we know that there's no biology to support them? The answer was no. They have a seed bank that was just waiting to wake up, and we were confident that the microbes will wake them up. And we really approach weeds as covers. They are our partners in revitalizing dead soil. So this section also was heavily disturbed by equipment last year because they had to put in the new infrastructure for the trees. They had to dig to install their irrigation lines and place posts to prep for planting this year. So fast forward to 2023, so summer of 2023, we know there is soil disturbance due to the new infrastructure. They put in their irrigation lines, they put in their posts. So we were monitoring nematode populations from the very beginning to identify the effects of our implementation. So on the bottom left-hand side of this chart shows the minimum desired, if you go along that row, minimum desired for deciduous trees for each nematode group. Our initial analysis was done in April 2021, and it's labeled initial on the graph. 3 treatments were completed in fall or winter of 2021. Another analysis was performed in the spring of the following year, so that's April 2022. And there's a visible jump in the nematode population due to the moisture and our treatments. And the disturbance happened again at some point between April 2022 and May 2023, so over a year apart, because they had to put in their new infrastructure. So we're making a few speculations here, but you can see the significant decrease in total beneficial nematodes on the very right-hand side from the time that they treated the field in April 2022 up until basically summer of this year. So there was disturbance that happened between that time, and there's a huge decrease in nematode population. So Ian built more compost. That's how we decided to remedy the situation. He knew that's what we needed to get done. So Ian sent a sample of the compost he wanted to use to remediate the South Orchard because he just planted new young trees. So overall, awesome biomass and diversity in the compost pile. I was actually getting pretty tired of counting his nematode numbers because there's way too many of them in his compost. So in July of this year, I revisited the farm, helped Ian come up with a new plan to support the newly planted young trees in the South Orchard after disturbance. This is his new compost setup on the left-hand side. He's using different equipment materials, so it's more robust. He's not using those flimsy mesh anymore. We built compost together while I was there. We also needed to assess his sprayer to make sure it's feasible for what we were trying to do. So do we need to configure it to accommodate for vertical spraying? We wanted to make sure that no changes needed to get done. So we also looked at his liquid inoculants under the microscope. So a total of 3 treatments later, I think 3 to 4 treatments later, this is what they sent me. Actually, I think I skipped the slide. Oh no, this is what they, yeah, this is what they sent me on the right-hand side. It was just puff balls everywhere. That's what the South Orchard looked like on the left. And over 2.5 years later, diverse plant species on the soil, lots of fungal growth, as you can see. But the work doesn't stop here. It's actually, we're just starting. So big round of applause to Little Dog Farm, Kathy and Ian Finley. We'll continue monitoring the soil and the trees, making sure that we're moving in the right direction. So one of the real tests will be this winter. We want to see those young trees survive the frost. And also in the spring, we want to make sure that the insect pressure, if there are any, it's not going to be too damaging for the young trees. And thanks for having me here today.
Thank you, Joe. I can't wait to see future years as that property starts to unfold. Fantastic, exciting. Okay, so before we jump into our Q&A section, we're going to have a quick video describing the Kickstart program. So we have this Kickstart promotion happening right now. And let me go ahead and just jump to the videos, and so we can actually learn a little bit about that.
Some of the farmers working with Soil Food Web consultants around the world are seeing remarkable results.
Just within the first 3 years of working with these folks, they saved over $1 million just by cutting back on synthetic fertilizers, going from 200 units of nitrogen per acre down to 150 to even 50 units of nitrogen with the same, if not a better yield, which is really exciting.
If we look at the fertilizer savings across 1 hectare, we're looking at about $1,000 in savings. And then when we look at the increased revenue from the yield increases, we're looking at about $504. So the gross margin was increased by about $1,500. And then if you extrapolate this over 200 hectares, we're looking at about $300,000 over the course of any given year.
You can see the average again, 72%. And if you just take a few of those examples, you can take celery or you can take kale, you can see that we reach over 100% of yield increase.
Basically, the weight gain again was like 25% or more. The only complaint I got was the client wasn't used to obviously bailing as often because the crop is growing so much better and so much healthier. They went from 280 to well over 400 sacks an acre. So that's 42% increase, which is really impressive. I mean, dollars and cents wise, that's a big jump. The PEs were really amazing. They were really aggressive with some of the PE projects. 136% increase, that's pretty impressive.
If you're interested in joining the soil revolution, this is a great time. We are making the Kickstart Your Soil package available for $39.20. With the Kickstart Your Soil package, you can register for the Soil Food Web School's foundation courses with Dr. Elaine Ingham for just $39.20, saving over $1,000 through November 1st. You'll also get Stage 1 of the Consultant or Grower Training Program totally free, saving a further $15.40. That's 26 hours of mentor time dedicated to helping you make your own biologically complete compost and develop your microscopy skills to qualify as a certified Soil Food Web Lab technician. The total value of this package is over $6,500, for which you'll only pay $39.20, saving over $2,600. That's 40% off. At the Soil Food Web School, we are so confident in the quality of our courses, we proudly offer a 100% risk-free guarantee. Here's how it works. From the date of your registration, you'll enjoy a 30-day window with a no questions asked money-back guarantee on the total price you paid for our courses. The only condition is that you cannot have watched more than 32 lectures of the foundation courses. Your satisfaction is our priority, and we stand behind our commitment to your success. Finance options are now available, so you can pay at your own pace with a firm. Register today.
As you can see, big savings through our Kickstart program. So we hope to see a bunch of future new students and eventually see colleagues out there working and doing the good work. All right, so now let's move to the Q&A. There was some really great questions that came in. And panelists, just to let you know, go ahead and unmute yourselves so we can actually have a good dialogue here. And the first question is coming in from Richard. And we get this question quite a bit, which is, what has been the biggest challenge in convincing industrial farmers and ranchers to transition to biological driven farming and land stewarding, as well as a soil food web consultant? So panelists, who wants to start off?
I guess I will. I would say for the most part, we don't really go out and try to convince a lot of people. You know, it's like, you know, one-on-one anyways. Putting this information out there, like going, you know, to events and speaking, things like that, you're pretty much speaking to people that already have that little piqued interest. And, you know, really it's it's kind of a little bit like, you know, talking to a brick wall sometimes if you're trying to really forcibly convince someone that this method is better. And so, you know, oftentimes we just put ourselves in situations where there's people that are already kind of interested in it, and people kind of then flood into those scenarios, you know, and they'll, you know, especially with farmers, is they want visual, they want to see it happen. So it's like, if you can get the one person in the neighborhood who's already a little bit interested in it and they're doing it, their neighbor's going to look over their fence and be like, well, what are they doing? Or they're going to talk, and that word's going to spread word of mouth a lot better than, you know, knocking door to door trying to convince somebody of it. And so I don't know, I think really the best way to do that would be to, yeah, you know, just put yourselves in environments where there's already people that are interested in it at least a little bit, so that you're not really fighting against that wall.
I bet, Cory, you probably your neighbors are paying attention to you, right?
They are. Yeah, it's been it's been really exciting to see, because we're right in the middle of town. Everybody gets to drive by. And one little experiment we did this year was I an alfalfa field that we went in and we planted some triticale in. We harvested the triticale and then no-tilled in corn. And it was really funny because that corn started to grow and everybody, what is that? And then, you know, the corn's 6 foot tall and then the corn's 8 foot tall. And they're like, what are you doing? And so it was this little experiment. I didn't want to have to look at you guys when you drive by. So I wanted to be seeing the rest of the stuff we were doing. But what was really exciting for us is we went in and cut rows in it for electric fence, and we're going to graze our cattle. So it's, you know, allows us to sell the hay we produce and then keep the nutrients on our soil. And ultimately that's our goal, is to get to where we're not selling hay, we're actually using the beef to cut our grass, and getting rid of this equipment that is very expensive to operate and maintain, and moving to a system that is more sustainable. So, you know, the question isn't, hey, Cory, what are you doing? The question is, hey, Cory, how can I do what you're doing?
Right.
And we have a lot of people that come. I mean, I don't know if a lot of people, if you guys know this, but every day in the summer, I have a class at 8 in the morning. And some days we have one person show up. Some days we have 30, 40 people show up. And they just want to tour and look at our compost and tour our fields and see everything. So, you know, I think that's the real answer. And like Casey was saying, is put yourself in the community and talk about what you're doing, as opposed to everybody else. You know, the conception of farmers is that you're trying to hide it from your neighbor. And what I've found is that's exactly not the case. Everybody wants to help you. Everybody wants to learn from you as well as you learn from them. And you know, a friend of mine made fun of me. He says, yeah, all farmers do is drive around and talk to each other. And I think he's kind of right. You know, if you're in a tractor, you're on your phone talking to another farmer about what he's doing. And so, you know, I don't see it as a challenge. I see it as a way just to communicate. And, you know, now it's giving me a lot more avenues to communicate.
And I think it often takes that first year of making your compost, figuring out in your soil, small farm of some kind, and determining what the biology is that has to be fixed, that you've got to put things back into that soil to make sorry about this throat, to make these organisms do jobs for your plants. And we can go into the mechanisms for all of those plants. Exactly how is it that nutrients are being improved, that disease is no longer attacking the plants you want to have grow. You don't have the weed species in there, you know, but as we were just looking, sometimes the weeds can be doing a useful purpose. You've got to get that soil surface covered. So all of these things, and you know, present them in a simple way. Have, you know, something on the main drag where you've got, here's the chemically grown, here's the biologically grown, and maybe a couple little differences in the things that you trialed, since it is a trial situation. And usually you get a significant number of people coming over. You don't want the whole town to come at the same time. You want to build up your clientele.
Yeah, I think Joe showed kind of a good example of that, which is we're really degraded land, and just within 1 year period of time frame. I mean, people had seen that land, in that degraded state, and then all of a sudden 1 year, it's like, wow, that's a huge transformation. That's going to get people talking. What are they doing over there to be able to drive that? That's just crazy. One thing I would just add to this, in case he's right on, normally when a client comes to us, they're coming to us for a reason. They have challenges that they have not been able to overcome. Their financial, picture's not looking great, in the agrochemical space. And so the farmer or the client is already in this mindset of, I want to make a change. Where I find sometimes the really challenge to be is in the whole support staff supporting that farmer. You've got the agronomist, you've got the fertilizer specialist, you've got the, irrigation specialist, the farm managers that have really been doing, the agrochemical way of farming for quite some time frame. And their exposure, experience, and their desire to move to a biological paradigm may not be there yet. And so one of the things that I will tell a lot of my clients is, you may be in this space, but we need to get your team in this space as well to understand what we're trying to do and why we're trying to do it. And so we'll usually try to do some kind of biological primer, an hour or 2 hour talk with their staff to really get everybody understanding, how that transformation is going to work and why we need to be able to foster good soil health and soil biology. And so once you can make that switch and you get the team behind you, then usually things will really start to move. Okay, panelists, anything else you guys want to add to this one? All right, we've got a compost question. This one's from Jay, and Jay asks, by learning the compost portion of the soil food web, can I do it for myself in a small backyard? Question number one. And question number two, how long from start to finish does the compost take to be ready to use it? What say you?
Okay. Yeah, you can definitely make enough compost in your small backyard to treat all your friends' lawns and your lawn as well. One yard of compost is what, 3 by 3. So you could do, as long as you have double that space to turn, you could do that in the backyard. But you don't even have to do thermophilic compost. You can do a worm compost. And if there's a book out there called Worms Eat My Garbage, I believe is the name of it.
Mary Applehoff.
Yeah, the amazing Mary Applehoff. And she talks about people having worm bins like literally in their house, like built into their furniture. Because if you're keeping it proper, right, like there's no smell to it, there's no flies coming, so you can actually make compost in a very small area. And it's depending on, if you're talking about thermophilic, it can be finished as quickly as 2 months, but often it'll take up to 4 months to really get to where you want to be.
Great. Anybody else want to add?
Yeah, so can you do it yourself in a small backyard? Yeah, I'd encourage you to come and take the foundation courses so that you really get the understanding of why these steps are taken in the production, even though it's a small backyard, in wire cages sort of thing, and getting all of the organisms into that compost, going out and getting more diversity all the time, adding it to your piles before you get them started. And how long from start to finish does the compost take to be ready to use it? Well, if you follow all the instructions and you're doing a pretty good job, it's only 21 to maybe 25 days. If you want to, kind of sit back a little bit and let things go a little bit slower, then yeah, it might take you 28 and a week, a month and a half, something like that. It's kind of up to you how much effort and energy you want to put into it. So, we always push. I've got to make more compost. I got to make more compost because we keep adding more property. So let us not scare you off from, I've got to have all this compost and everything going. The compost is there to bring those organisms that you're missing back into the soil that you want to grow in. And it can do a really good job. And it's a bit of you're the captain of your own ship, and you can arrange for these things to go faster or slower. When you come and take courses from us at the school, you'll have people, lots of different people to talk to. And so you'll be able to kind of pick their brains and what do they have, your mentor for the different steps in the process as well.
Yeah, one thing I would add is even when we are approaching, wanting to make compost on a large scale for a client, let's say we have a large farm, they want to make on-farm compost. A lot of times when we're teaching them about making compost, we'll actually have them make small batch hand-turned piles, simply because it allows them to be very intimate with the composting making process. When you're out there managing a small, like a hand-turned pile, you're constantly smelling, feeling, turning yourself. You get to experience and internalize what a good compost, as a good compost develops. And so, even though they might be making large scale compost, 6 months down the road, we'll still have them make small hand turned piles initially, just to kind of get that experience. Panelists, anything else you guys want to add? Okay, let's move to the next question. This question is from Eric, and he says, I love composting, but have concerns about pathogens like Pythium, bacterias, viruses, viroids, etc. Some of these can be very heat resistant in high amounts. When composting, especially with ingredients not local like food, other garden waste, etc., how can we be sure to eradicate 100% of them and not 99.99%? For a big farm, you can compost from your own farm, but if you import compost, how can you be sure it is safe?
Well, you really do have to make certain that the people are following the right protocols. And so typically you're going to be asking for their sheets that show the time of turning, the temperatures, what was the starting material, all of those typical things. And then you want to, in the temperature cycle, you want to make certain that everything got above 131 degrees Fahrenheit for at least 3 days. And then you want to repeat that 3 to 5 times to make absolutely sure that those disease-causing organisms, the virus, the root-feeding nematodes, things like that, are not going to be able to survive. And so it's very important to be measuring that temperature and make sure that every part of that pile gets into that hot middle where you're up above 131 degrees Fahrenheit for a full 3 days, or 145 degrees for 48 hours. So it depends on how rapidly you're getting really hot. And so go take those classes from the school. And we've looked at over 800 compost piles, for example, and what were the results. And out of the 800, there were 0 that had a detectable human pathogen in it. So if you understand your conditions, you can be successful every time. But if you're not paying attention, you're not doing things quite right, you will then see those disease-causing organisms in your compost using your microscope. So right there is another way to determine whether you can, guarantee practically that it is going to be safe.
And Elaine, I think it's also about how you maintain the compost too, right?
Mm-hmm.
You can make great compost, and then in the maturing stage, you can really overwater it, make it anaerobic, cover it with plastic and all those other kind of things, and start to change the properties from aerobic to anaerobic. And so I think it's not just when you make it; it's also how you maintain it.
Yeah, how you store it. And so you want it, in there 40 to 50% moisture, but no more. Really important.
I guess I would also add too, is like in that question, you cannot be 100% sure you got rid of everything at all. What we're trying to do is basically build an immune system for plants, right? And so if you have everything in that system, your nematodes, you have that diversity in that compost, And when you put it out to that plant is going to be healthy. So it is going to have defenses against those things that could possibly be in your compost or even just in your soil, you know, like things like Pythium, that's floating in the air everywhere around you at all times. You're probably breathing a spore of it in right now.
Not to make you panic.
Not to make you panic. Yeah, exactly. But you have an immune system, like, or it doesn't grow inside your body, but they're everywhere. And so you can't be certain that these things are gone. But if you're healthy, your body can fight these things. It's like even when you send in samples of compost, like we have to test for salmonella, we have to test for E. coli. The E. coli numbers show up, they're just way below the threshold, right? There's a certain amount of E. coli you're allowed to have before it's an issue. So they're there, it's in there. It's just the amounts that they're in. And then, you know, it, like we said, if you don't treat your pile properly, okay, now you've got it super wet, things are going anaerobic. You've just created an area where now that E. coli can bloom, right? And so, you know, doing it as in, you know, in the realm of the best that you can when you're making this thing, there are steps to follow, but yeah, you're not going to get rid of everything. It's just making sure that everything's there for the plants to be healthy.
There's a resiliency in having a diverse community of microorganisms. So when we're talking about using the microscope to assess the diversity, if we're seeing every time we move our microscope, you know, different colors, shapes, and sizes of fungal hyphae, if we're seeing lots of different shapes of amoeba and flagellates, if we've got lots of different nematodes, there's something to that. When you see that when you see that diversity, you're getting a protection from that. So this is a lot of like, we're hedging our bets. We aren't certain cases, right? But by investing our energy into the community of organisms in the soil, that's going to reflect above ground and in ourselves, right? Like the more diversity of microorganisms that we actually get to be in contact with every day is going to help our immune system. This is a big cycle of life, and we're influencing it with this work all the way up to ourselves.
Yeah, and I don't think 100% eradication is really the goal. It's making, you know, where you're making it so that the pathogens do get killed off for the most part, and you have, like you said, a massive diversity that's going to suppress any kind of expression of those particular pathogens that are there. All right, panelists, anything else you guys want to add to this one before we move on to the next one?
I do. I don't know why I have to say this, but like fusarium, right? Fusarium is a huge issue in plants, but in the right proportions, fusarium can actually be an endophyte and live in your plant's roots and use it to defend itself against other types of fungi. So, you know, also they're not all bad. Sorry. I don't.
Yeah, not all fungi are bad. Most fungi are good.
Even pathogens aren't bad
Some
necessarily.
bad guys can be good guys sometimes.
Yeah. We make it in a situation where, yeah, okay, maybe a few might be in there, but they're being outcompeted, they're being inhibited, they're being consumed by all the beneficials. And so if we have a massive amount of those beneficial organisms in there, I don't think you have to worry ever.
Agreed. Okay, next question. This one's
Sorry,
from
Brian.
Oh, go ahead,
I have
Joe.
something to add.
Yeah, by all means.
Talking about importing compost, how can you be sure if it's safe? I think it's good to have those conversations with the composting facility as well. What kind of ingredients are they using? How are they managing the compost piles? Or maybe even if they're open, maybe even submitting their sample to a lab tech that knows how to check for those microbial communities that we're looking for. So yeah, I mean, that's real. And also using your senses, right? Talking or smelling the compost, making sure that it's not black, doesn't look like putrefied organic matter or anything like that. Those are some ways
No
in which
clouds
you can,
of
yeah.
no clouds of you know, flying annoying flies and ants,
yep.
and yeah, those
Yeah.
are direct draws.
Yeah, and your body knows. I think our bodies know what materials are, you know, we prefer over others. So
Yep.
that's
And
all
if
to
you
add.
don't know now, you will know pretty soon, after you've made, you know, your second, third compost pile, and have people around you that know what the proper smell is, know what it should look like, know what the aggregate structure should be. Then you pick it all up. It's a pretty fast process.
What's really funny about that too is once you've either made your own compost or you've worked with really good biologically complete compost, I have a lot of clients experience this when they now go out and try to source other forms of compost, like some bulk compost for organic matter. Boy, they're like, there's no good compost out here. What is going on? The vast majority of compost I get to go out to the compost lots is just garbage. And you're right, that we've got a long ways to go to help start making really good biologically complete compost. But I'd have to, one other thing I would just add to that too is you have, without a microscope, you still have the good skills. And Joe was mentioning this, it's your eyes, your nose, and your hands are good tools to help you kind of pre-qualify what you think might be okay compost.
Or right away it's bad.
It's bad.
Do nothing before you go into a bathroom and wash your hands.
Exactly. Yep.
It'll smell like said manure.
Yeah.
If it has a chicken vibe, no go.
Yeah, no go. Exactly. Yep. All right, we've got time for one more question, and this question is from Anne. And Cory, this one's gonna be geared to you just right off the bat. But would you explain more what cleanup means when you put fungal tea on your fields? And it could be fungal tea or compost extracts, liquid amendments.
Yeah, extract. So we bale graze, and for people that don't know what that is, that's in the winter time. We'll spread some seed that we want to reseed in that field underneath of a round bale of hay. We'll put that bale in the field, and then when it's time, and we do this in the fall before the weather gets bad. Purpose one, we're not running a tractor to put hay out for cattle, and it's there if we need it, but if we don't have to use it, then we don't. But when we do actually put cattle in there, of course, they don't eat all of it. They like to lay on it, and they go to the bathroom on it. And at the end of the season, or at the springtime, when we want those fields to start growing again, it has a layer of matted down hay. And of course, that's blocking the plants below it from coming out. And we need to clean that up. So in the past, farmers would drag their fields to clean up in the springtime. We did that, and we found that as we drug over these piles, it just made a bigger mass. It rolled them up into even a larger pile. And found and it wasn't by chance. I knew what we had in our compost, and I said, let's see what happens if we put a massive amount of biology in here, and if we can really have it go to work. And in the springtime, when it starts to warm up and everything's wet and the conditions are right, the biology flourishes. And they're the lignin degrading microbes that we talk about. And so that's the purpose. The purpose is just to break down that lignin left over from that winter's hay feeding. And last year was very successful for us. It limited the amount of time we had to spend cleaning up the fields. And really in those areas, the cows eating on that bale are putting down a lot of fertilizer. And what we saw this season is we took our first cut, everything looked good because conditions were good coming out of winter. But second cut, is that grass would grow back, where everywhere that round bale was sitting, it was double the height, double the production in those areas. And a lot of it had to do with the seed we put under it, but getting it cleaned up. And what you'll find in farming is you have to do these processes, and you're always managing your finances. How do we do it as cheap as possible? Well, we have this material, and if we don't have to start a tractor to do that, or something else, another implement to move across the land, and we use biology, then by all means, let's do that. And it's a tool that we use, and it works quite well.
Fantastic. I'll use another example. We use the same kind of process in almond farming, where because of the way they have to harvest the almonds, at least the way that the equipment's been set up, they need bare floors to be able to shake the nuts down and then sweep them in the middles and then dry them in the middles. But we need to be able to get some cover on the soil for a good amount of time that we can. And so we'll grow cover crops in the middle rows of the almonds. And we do a crimp and roll in springtime frame. So basically we'll roll down that cover crop, and then we'll terminate it into a thatch. Well, we can't have that thatch in there by the time we get to harvest time in fall. So we spray a compost extract over the top of that thatch, that crimp and rolled. And by the time we get to fall, it's pretty much broken down. That material has been consumed by the microorganisms, turned into humus, and now reincorporated back into the soil profile. So it is a really effective tool to use microbes to be able to clean up those fields.
We do the same thing with lawns. here in Southern California, it actually doesn't get too cold, but people have grass all year round and they're cutting it very, very short, and we get that layer of thatch that builds up underneath. So we're always coming through and aerating and adding the compost extracts, and it breaks it down quite quickly.
Yep. Yeah, biology, such wonderful partners in the work that we do. Okay, panelists, anything else you guys want to add before we close?
No.
It was a really fun webinar. I liked everyone's presentation.
Yeah, this is fantastic. I thought this webinar was one of the best, in my opinion.
I think we got the topic kind of focused in really well, and it was easy for everyone involved to show their data.
Exactly. Yeah, and Joe and Cory, fantastic data on some of those projects that you guys were working on.
Yeah, I loved your before and afters. Those were awesome.
Yeah, fantastic. Yep. Okay, so I've got a little bit of housekeeping to do here. Give me 1 second. Let me jump to this slide. And all I want to just talk about here really quick is our upcoming webinar, which is going to be 11 a.m. Pacific on Wednesday, November 1st. And this is going to be Dirt to Soil, Transitioning to Biological Farming. So we're going to hear from, again, more examples of what we do out in the field to make those kind of transitions. And as always, you can watch the replay of this webinar plus the one that we had last week. So go out to webinar.soilfoodweb.com. And other than that, I just like to give some great thanks. We have a wonderful support staff of folks that set up and promote and do all the work of getting these webinars put together, plus also a team that maintain getting the technical, getting the webinar set up and running our questions and making it easy for us as panelists to be able to do the work that we do. So thank you very much for all the support staff behind the scenes that do a great job. And panelists, awesome job. Again, great conversation. Thank you so much for being able to share all that wonderful information. And the last thanks I like to give is to the audience. Without you, we wouldn't be here doing this. So thank you for attending and showing interest and hopefully getting yourselves excited to be able to do this kind of work.
And you and Sammy and the whole support staff do an incredible job every time.
I agree. Thank you, everybody. All right. Well, we'll see you soon in our next webinar, November 1st. Thank you, everybody.
Bye.
Ciao.
Bye.
Bye, everyone.
Bye. Thank you.
Don't forget to click that like button, subscribe to our channel, and ring the notification bell to stay updated with all our new videos.